everybody, in this video we're going to look at independent assortment, which is one of the ways that genetic variation is introduced during sexual reproduction. So this is what we're trying to answer. If we look at gametes, then gametes are different from one another, and we want to know why, if you look at different gametes, they have different combinations of chromosomes in them. So different combinations of maternal and paternal chromosomes. So if we start off by looking at a cell or a nucleus within a cell, and we're just going to imagine that this particular cell has got two pairs of chromosomes. So we're using the red to represent the paternal chromosomes, so they originally came from the father, and the blue there could represent the maternal chromosomes, or the other way around, it doesn't matter. But what you can see is we have got I'm using the length to represent the homologous pairs. So that chromosome there, the longer one, its homologous pair is here. So that's one homologous pair. And then we've got a short one. There's a second homologous pair. So at the beginning of meiosis, the first thing that would happen is that these chromosomes would replicate. The DNA would replicate. And then the homologous pairs would pair up. So during metaphase 1, the homologous pairs would line up along the equator like this. So there's one homologous pair, there's the other homologous pair. And then during anaphase, these pairs are going to be pulled apart to the poles of the cell. So if we think about the way that these are lined up, the homologous pairs are lined up here. On this side, on the left-hand side, are the two, what we'll just call them red chromosomes, and they're going to get pulled to this pole. So this cell here will have one red for each homologous pair, and then the other cell would of course have the blue chromosomes from each homologous pair. And then the second stage of meiosis would take place. So this cell here, the chromatids for each chromosome are going to separate, and we end up with our two daughter cells, and the same for the other one. So this daughter cell here has got one of each homologous chromosomes in it, and it has the two red ones. That would be the same. This would have the blue, and that would be the same as well. So we've got two different types of gamete here, this one and these ones. But this isn't the only possible way that we could end up with chromosome combinations. Because if we go back to the beginning, so we've got the same cell, okay, the same two pairs of chromosomes. They replicate in exactly the same way. But this time, when they line up during metaphase 1, they line up in a different order. So this homologous pair is lined up in the same way here as it was over here. But this second homologous pair is lined up the other way around on the equator, as you can see. Okay, over here it was red on the left, then blue, and here blue on the left and then red. So what that means is when anaphase takes place, the chromosomes get separated in a different way to the way they got separated over here. And already you can see that we've got variation. So this cell here has got one red and one blue, one blue, one red. So therefore when we then go through meiosis 2 and we look at the gametes that we end up with, what we see is that we have different chromosome combinations in these cells compared to what we have in these cells here. The only reason that we've got different combinations is because during metaphase 1 the homologous pairs lined up differently on the equator. Independent assortment is called that because it's saying that the different homologous pairs are independent from one another. This pair can line up in a certain way, in this case red-blue, and this pair lines up independently. So it could line up blue-red or red-blue. The pairs are independent of one another. in this cell you've only got 2 pairs, but if you think about humans, we've got 23 pairs. So if you think about the possible combinations that you could have where each pair could line up like this or like this, it leads to an incredibly large possible variation in terms of those combinations, and therefore in terms of the combinations of chromosomes that you get in the gametes, it's absolutely enormous. So that's independent assortment, and that's how we end up with gametes with different combinations of chromosomes in them, which is one of the ways that genetic variation is introduced during sexual reproduction.